So this slide is important to me because it's the slide I use when I give professional research talks. So part of my job as the head of the lab is to explain to people the research we do, and the primary audience I do that to are other scientists, and in particular other scientists that decide about funding. So all of our research is funded by taxpayers. So we get grants awarded by funding bodies that are established by the government. For example, that logo on the top left is for the BBSRC, which funds bioscience research. Another one will be the MRC, the Medical Research Council, which funds medical research. And so my lab is funded by both of those. And the way this works is you come up with a project and an idea. It could be anything to do with biology or medical research, and then you have to really sell that idea to other scientists who look at your work, they peer review it. And based on what you've done before, and based on how things are going now, they'll award you money or not. And it's usually not, right? So it's very hard to get money because everybody wants to do research, but it's fun. So this is the slide I use at the moment at the beginning of my talks. And for those of you that have been thinking about the worms, hopefully this makes some sense, right? So the title is Establishing and Exploiting Novel Model Systems, in this case, the worms. Model systems being any animal which you can use to do experiments with and study. For understanding aging, cancer, and regeneration. So I've got all three in there, all three big things that you probably see. If you pay any attention to pop science or newspapers, you probably see aging come up a lot, cancer come a lot, diseases of aging, things like Alzheimer's or Parkinson's, and regenerative medicine, the idea of using stem cells or nanotechnology or robotics to help people recover from injuries, all different types of injury. Okay, so that's what we do. So why is this important to us? Why is it important to our society? At the moment in the UK, there's a massive move towards trying to understand aging, and it's broadly called the healthy aging agenda. And the reason for that is more of us are going to live longer and longer. So average lifespans just keep going up. And many of you in this room will live to 100, and it won't be surprising. If I lived to 100, that was going to be surprising. But you guys who are about 20 years younger would not be so surprising statistically. It'd be like 50-50, you're going to get to 100, for example. So if you're going to live longer, that's great, that's wonderful. Some people might think that's a good thing. The problem with this is, as you get older, your ability to lead a healthy lifestyle decreases. So you probably have grandparents, I certainly do, and I've watched them grow older. So when I was your age, my grandfather could play football with me. Now that I'm my age, he's still there to football with me, because I've lost a lot, well, I've lost a little, but he's still in good shape. But anyway, the point is that we all get older, right? So he's not a good example. But as you get older, a lot of chronic and acute illnesses, so things like just getting ill and not wanting to recover from an infection, or diseases that we know are associated like dementias, which are the most popular ones in the media at the moment. Cancer is a disease of aging. Your chance of getting cancer increase with age. Okay? And then there's just a general, as I said, your ability to do things decreases. Your body's ability to recover from exercise, from anything, just gets slower and slower. Okay? So this has really huge economic and social impact, because we have all these really wonderful old people who, sadly, are not well enough to work and contribute in an active way. And so retirement ages, you may know, for example, have gone up recently, very recently, because people are healthier longer, but it's still going to be a problem. So if you all live to 100, that's great, but what are we going to do? Who's going to go to work, do stuff, simple things, right? So in order to try and alleviate that, there's a whole bunch of things we're trying to do. So in a nutshell, what I just said means that you may live longer, but it doesn't mean you're going to be healthy. So if you live longer and are unhealthy, that's a problem, and that's what we want to try and tackle. There's lots of ways to do that. So the first thing is very practical. It's just have healthy lifestyles. Don't smoke, don't drink too much, don't eat bad food, exercise, be health conscious, be health aware. I mean, don't go to ridiculous extremes. So as you get older, you'll be healthier. That will help, right? The second thing are direct medical interventions, right? To alleviate diseases associated with aging, whatever those treatments are. But the ultimate goal as human beings, we tend to always try and engage technology to solve our problems through history. So the ultimate goal might be to use technology and science to understand aging so we can interfere with it. Okay, and that's the goal of the research area I'm in. I'm not sure it's my goal, but it's certainly why we get lots of funding. Okay, is to try and understand aging as a biological phenomenon and then intervene. Okay, we know why you age, and we can stop it, slow it down, maybe even reverse it. Okay, so that's the ultimate goal. Okay. So how do we go about that? So as you know, because you're biologists, a lot of what we are depends on how the cells in our body behave. And the cells in our body are underpinned by molecular information in the form of DNA and proteins in our cells. So at the bottom of aging is this idea that the cells in our bodies age, they're unable to replace themselves. And that leads to aging. Simple idea, and it's probably true. Okay? So we'd like to spend some time as scientists, I mean the whole group of scientists that study aging, understanding what the genetic causes are of aging. But also what are the environmental causes of aging? Things like things you do that make you age quicker, for example. Smoking is the most obvious one. People who smoke, well, they age quicker, right? On average. There will be some people who keep smoking and live to 100, but most won't. Okay, you'll get ill. You'll die younger than you would if you hadn't smoked. So once we've identified those causes and we've described them, we're sure what they are, and we're not at that stage yet, I would argue. You know some of them, the genetic causes and the environmental ones, but there's probably a lot more than we know. What approaches can we take to alleviate those? Okay. Does it make sense so far? In some detail. We know a lot about aging, or we think we do. So I'm a molecular biologist, so I look at things from the point of view of molecules. Okay? So some of these you may have heard of, right? Some of them you won't. So a very famous one, Nobel Prize is awarded for this, for the discovery of telomeres. So at the ends of your chromosomes, you have structures called telomeres. And each round of cell division, they get shorter. When they get to a critical length in a particular cell, that cell stops dividing. So you can see how that ties into aging, because if that cell can't divide and you have some damage to a tissue and you can't repair it, that's going to cause aging, right? In a sense. Okay? So telomere attrition is an example. Another example which I think is really easy to understand is stem cell exhaustion. And of course you can see that stem cell exhaustion and telomere attrition are linked to each other. Stem cells might over-replicate or replicate to the point where they can't do it anymore because of this telomere mechanism, right? So all these things on here are different molecular mechanisms associated with aging. Okay, I'm not going to go through them all. And once you have those, you can then think about how you treat them or how you alleviate them. So all of these different mechanisms affect us at different ages, or can affect us, or start to affect us at different ages. So for example, your telomeres start to get short in middle age, so around my age, telomere attrition starts to kick in. So is there a mechanism which we can reactivate the enzyme telomerase and start putting these ends back on so our stem cells can keep dividing? Okay, that's an obvious example. Right? Another reason that we age is because of inflammation in old age. Our joints wear out, we get inflamed. Our own immune system essentially, over time, attacks our own body. Can we undo that with anti-inflammatory drugs? So people already take aspirin for heart disease, but perhaps it also helps with it, with aging by reducing inflammation, for example. Okay? So the bit of aging research that I'm really interested in is this relationship then between what we call chronological age, so real time, how long are you alive for? Okay, so that's specific to each organism. So in our case, our lifespans are quite long, other animals have quite short lifespans, and it's linked to replicative age. So what I mean by that is the number of times each cell in a body, and that cell can be contributing to any tissue or organ, is divided. Okay, and there's a relationship there. So for example, if you can increase replicative age, can you increase chronological age? Alternatively, if you can increase the time you can be healthy in between these replicative divisions, can you actually, with the same replicative age, increase chronological age? And of course there's safety issues here, because if you have cells dividing out of control, you might get tumors, you might get cancer, right? So when we think of these things, we actually think in terms of stem cells, really, because they're the things that are doing this. So in you guys, your stem cells are fine. They do the things proper stem cells should do, which is, this red arrow here just means they self-renew. Stem cells have to help to make more stem cells. They also have to give rise to healthy progeny. So progeny are all the cells that go and do stuff. So stem cells often don't do much. It's their progeny that go and do stuff, muscle, nervous tissue, etc. Okay? And this is just a list of different things that can go wrong then during aging. You can have an interruption to the self-renewal process. You get a depletion of the number of stem cells, which means you have less progeny, which means as an organism you don't function properly. You don't have enough of any particular cell type. Stem cells can go wrong and produce the wrong types of progeny. So instead of producing the right types of cells, you get an error or a mutation in the stem cell that starts to produce the wrong type of stem cell, the progeny cell in the wrong place. So you can imagine, I mean, in a really simple scenario, when to produce muscle and you start producing fibroblasts, which are muscle, you're going to damage the function of your muscle tissue. Okay? And that's going to lead to aging. And so on and so forth. And perhaps the worst one is here, is these stem cells here cycling out of control and become malignant and are going to cause cancer. Okay? So that's perhaps the major issue, and perhaps might be the major disease of aging. Okay. So as a scientist, I came to this problem thinking, well, I really am interested in this, and this is what I might want to spend my time doing. So how do I tackle this? And so I was probably 5 or 6 years older than you are now, and I had done a degree, and I'd even done a doctorate, and I still thought I didn't know very much. That's because I didn't, and I still don't in some ways. And so at the time I came to this problem, people used 3 major model organisms to study aging. The top one you recognize is a mouse, and a mouse, in research terms, from molecular biologist or developmental biologist, is a proxy for us. Because in evolutionary terms, we're quite close. And so mouse has all the same organ systems and structures that we do. And so when we find out things in a mouse, the hope is, it turns out it's not always true, but the hope is it's applicable to humans, or at least the amount of work required to understand it in us has been greatly reduced. Okay? That's a good model system. The downside of mice is that they're quite complicated. So if you imagine trying to study a system for the first time, or study a problem for the first time, starting with something very complicated isn't always very useful. You might want to strip it down and start with something simpler. And so for that, people had used 2 other model organisms, invertebrates. First you've probably heard of, everyone's heard of, the fruit fly, Drosophila. Put your hands up if you've heard of fruit flies and fruit fly genetics. Okay, good. And the other is the nematodes, Caenorhabditis elegans. Put your hands up if you've heard of that. Might be down to 1 person now. Okay, yeah, or 2 people, right? Okay. So the advantage of these animals is they're small, they have very short life cycles, so you can do experiments very quickly, and they have lots of offspring. And the lots of offspring, called fecundity, is very important, because we can actually go and do genetics. So we can make a change or induce a bunch of random changes in an animal. And because that animal has lots of offspring, we can easily assess the effects of those changes. Okay? So what you would do classically, when I say classically, I mean 20 years ago, that's classically for me, is take these animals and just expose them to mutagens, if you're interested in aging, and see how long they live for. And you look for offspring that live longer than average, or much longer than average. And the hypothesis is that you've made a change to the genome which leads to life extension of these animals. Okay. So most of what we know about the genetics of aging, the genes involved in controlling cells and how they age, comes from that process of these 2 animals. And then being transferred into mice. And there's some genes that are found in mice too. And it turns out that in these 2 animals, one of the things that really regulates their lifespan is nutrition and how much food they're getting. And if you reduce signals that indicate nutrition, or actually reduce nutrition and actually feed them less, their lifespan gets extended. Okay? We can talk later, perhaps if there's time, about why that is. Okay? Right, so I came to this and thought, right, so I can learn about Drosophila and see how to do it. I'm not going to work with mice. I tried working with mice once. Maybe very unhappy. That's just me personally. lots of amazing research done with mice, and it's incredibly important for all of us that continues. But personally, I always just found it hard. A lot of my friends don't have an issue, but I've always just... I don't have such strong feelings for worms and flies. Anyway, that's just me. So my options then were really these two. And I thought about it for a bit, and I thought about where I could go to this, where in the world I could go and do this. Actually thought about working on fruit flies more seriously than C. elegans. I started off with this one and thought about this one. And found that I wasn't actually studying aging. Two reasons for that. One is I got sidetracked and interested in other things. But two, I thought it was really strange that you would study aging in two animals that are really short-lived. So C. elegans lives for a few weeks. Drosophila can live for a month and a half, 2 months. It's not very long. And so if I study aging in these animals, what am I going to learn about aging in humans? I'm not sure. I wasn't sure what I was going to learn about. Not that we haven't learned about aging in humans. I just thought it was a bit strange. But I thought more about our own biology. We're chock full of stem cells. Human beings, right? So you replace your skin every 3 or 4 weeks, the lining of your gut every few weeks. I believe you replace all the nephrons in your kidney every 7 years. So different tissues in your body turn over at different rates, but they're constantly doing it. The brain's an exception, but nonetheless, we are regenerative organisms. Stem cells are always active doing stuff. We wouldn't be able to live for so long if we didn't have cells to replace the dying cells. Okay? These animals, not so hot on stem cells. C. elegans is post-mitotic as an adult. So as an adult animal, there's no more cell division. It's done. And this number of cells is fixed. In fact, one of the reasons it's so famous is because its number of cells is fixed. The adult animal has just over 1,000 cells, and that's it. No more cell division. Drosophila as an adult, slightly different, has no stem cells except in the gut, which makes sense. They eat a lot of food, they encounter a lot of pathogens and bacteria through the food they eat. So they have gut stem cells, adults, but nothing else. And they don't live very long. So I'm thinking, well, are these the right animals? Okay. I'm going to skip that for now. Oh no, I won't. I think we can go into this. Right, so if we go back a step and we think about, or you guys think about, why do animals age? Why do we age? Why do we live forever? What's the reason for that? So we can explain that from different points of view. We can describe the mechanisms as to why we age. We age because there's no stem cells. Well, the stem cells aren't working properly, for example. Or we can think about the historical reasons for why we age. The evolutionary reasons as to why we age and die in the first place, okay? So any of you that have encountered hypotheses like the selfish gene, or read other kind of hot science books about how genes are passed on from generation to generation, you have this idea, I hope, that the only real reason we exist is to pass our genes on. We're machines that pass on our genes. That's an evolutionary explanation. Okay? The scientific evolutionary explanation for why we do that. So then aging might be wound up in this process, right? So then we can ask the question, if that's true, how does natural selection result in animals that age? Why would it do that? Okay? In some sense, it doesn't make sense. We're alive, we've got the genes in us, why don't we just keep going? Why do this process of reproduction? Why would natural selection select that in so many animals? So one idea is, well, maybe aging itself is adaptive, and lots of people have played with this idea over the last 100 years. And maybe aging and dying makes space for the next generation. Yeah? So maybe we die and make space for generation, and that's good for our genes, which pass from generation to generation. For various reasons, that argument doesn't work so well. A good reason, or a reason I think does work scientifically or logically, is that the strength of natural selection decreases after reproductive maturity. So once you've had your children, whatever animal you are, it doesn't matter what happens to you, because your genes have been passed on. Okay? So then, natural selection acts to get you to the point where you reproduce, and you have vigor and strength and vitality, and then you reproduce, so it doesn't matter anymore, you can just die. But that doesn't explain why aging happens. I think you have to take that a bit further. One way to take it further is this idea that actually our bodies, our soma, somatic cells, as opposed to our germline, are actually disposable. They don't matter. You have to only invest into the point of reproduction, and after that, natural selection doesn't see it anymore. Okay? So from that there comes an even more interesting idea, which I really like, which is this idea of negative pleiotropy or antagonistic pleiotropy. And the idea here is that the genes that make you great at reproduction, or make you competitive to reproduce, have, by chance, a negative effect when you later. So natural selection cares that you're reproductively successful, and so it gets you to that point. But some of those genes that made you very successful have a negative effect. You later actually drive the aging process. And that's just by chance. That's a pleiotropy. So when a gene has more than one function, you call it pleiotropy. So this gene has an early function, which is good because it helps you reproduce, but a second function, which is bad. But that doesn't matter, because natural selection doesn't care about that second function. It doesn't see it because you've already reproduced. Okay. So I hope that those ideas make some sense to you. Okay. So having thought about that, let's be a bit more practical. So how would you actually treat aging? So you could slow down the aging process. Okay, you could imagine drugs that slow things that go wrong down, so mutations, for example. You could reawaken the processes that counteract aging. So that's what regenerative biology and regenerative medicine aim to do, actually, is through all kinds of different technologies try and do this. And so, from my point of view, was to take the model organism approach. Because it turns out regeneration as a phenomenon is actually really widespread among animals. It's just difficult, it has been difficult to study. Okay? And some of these animals that regenerate, it turns out, may not age at all. Okay? So here's a kind of pictorial tree of the animal kingdom. So we're up here in the mammals, and you have other vertebrates here. And these are all examples here of animals that regenerate. So in the vertebrates, with zebrafish, it's a famous model organism that regenerates a lot of different tissues. Salamanders, or axolotls, can regenerate their limbs, their tails, their eyes, bits of their brain, some of their heart tissue. And so they're the kind of champions of regeneration up here in vertebrates. And you can just see that all these different animals, all the way through animal phylogeny, phylogeny being the tree of life, can regenerate. You might recognize this one here, so maybe which is the Planarians. And regeneration is used through the animal kingdom for different things. So here you can see limb regeneration in an axolotl. Here's a sea cucumber, which is eviscerating itself entirely in response to a predator attack. It's chucking out all of its guts. So it's whole intestine, the whole lot. And it's going to use and repel itself in the other direction. It's going to leave this as a meal for the predator. It's going to regenerate all its guts in the preceding weeks. If you've ever been to the seaside, been in a rock pool, and if you haven't, you should, because it's where you can learn some biology. I was lucky to do that recently with undergrads. I've never done it before. That was like last year I did it for the first time. You might find starfish are kind of limbs, and this one here is just regenerating some limbs. It's probably if you're being eaten, actually. Okay. And sometimes regeneration in nature can go wrong. Here's a gecko that has, through a process that we call apomorphism, thrown its tail away, probably again to try and get a predator distracted. And when its tail is regrown, it's regrown. There's two little tails instead. Something's gone wrong with the regrowing process. So that's not so good for this gecko. It's going to be very confused when it comes to balancing. And regeneration can be used as a form of reproduction. So this animal is an annelid worm, and it's using regeneration to actually reproduce. So in this case, you can see a new tail and a new head forming here. This will bud up to form a whole new animal. So it's an extreme form of regeneration. This animal can regenerate absolutely everything, and then uses it as a form of reproduction. Okay. So there is a lot of people who say, well, mammals aren't very good at regeneration, but there was a recent example published a couple of years ago in Nature of the African spiny mouse. So this mouse Looks like this. And one of the really clever things about it is that all this skin here, which is spiky and protective, is also very loose. And its main predator is are snakes. And snakes grab hold of this dorsal tissue, the skin on the back, and they pull. And when they do that, what actually happens is the skin just comes off with the spikes. And the mouse is fine. It can actually regenerate up to 80% of all its skin with the spikes. So this animal is incredible at regeneration. And you think, well, the skin's not a big deal. Actually, your skin is an incredibly complex tissue. Arguably, your skin tissue is as complex as anything that you would have seen in a Planarian flatworm in terms of numbers of cell types and the way they interact. So that's a pretty profound example of regeneration. Okay, but this is the model organism after thinking about what to work with. This is the one I chose. Enough of that. So that YouTube video is from a Japanese game show called The World's Most Amazing Animals. And as you saw, there was a number 1 in the bottom right, because the Japanese public had chosen this as the most amazing, or at least the game show had chosen it as the most amazing animal, because it could regenerate. And I thought that was as good a reason as any. If the Japanese think it's the best animal in the world, then I think we should work with it. Okay. Right. I'm going to skip that idea. So the work that you guys are doing in the lab, some of you are doing, started at least 200 years ago, probably more. The first person to study Planarian regeneration systematically was a woman called Harriet Randolph in the mid to late 1890s. She was a graduate student with Thomas Hunt Morgan, who went on to invent the laws of hereditary genetics using fruit flies. But Thomas Hunt Morgan started out with Planarians, moved to fruit flies when he realized he couldn't really study genetics using Planarians. And what Randolph did in this study is systematically just go through with a particular species of Planarian and do all the different cuts you can think of. All the kinds of ones that you were asking me about a while ago in class, and probably thought about yourselves. And she was able to show that basically you can cut these animals anywhere, and they'll regenerate. So in this example here, she's taken a tiny little sliver here, and it's regenerated to form a new animal. And she's basically used shading just to show what's the new tissue and what's the old tissue. And this was her kind of interpretation of that result. Before these studies, a guy called John Graham Dalliel in Edinburgh, had called them immortal under the edge of the knife. It's a famous quote, and a lot of people use it. You should always go back and check references if you're really interested in something, because of course it turns out he said almost be called immortal. And I'd agree with almost, and I'm sure you'd agree with almost, because they probably all died, right? So you try to actually do anything fun with them, they start to die. It takes some practice to keep them alive long enough. Okay. So you'd have seen them on the outside. This is what those worms that you're working with look like on the inside. If you haven't seen these kind of images already, on the top left is a stain of a gene expression pattern of a gene expressed in the gut. Okay? So it's a messenger RNA. You know what messenger RNA is. You know. Okay. Messenger RNA is the intermediate between DNA and protein. DNA codes for proteins, and the messenger RNA is the molecule in between that takes information from the DNA to make the protein. So we can use those messenger RNAs to detect where genes are expressed in the animal. And so on the left is the gut marker, and the gene that marks the gut. Here the nervous system, in green. And here you have, I guess, the things that everyone is excited about, which are what we call neoblasts. So these are the stem cells of these animals. And they're the only mitotic cells. So these are the same cells labeled for gene for cells undergoing the cell cycle. Okay? So stem cells in all animals have a similar appearance in us, in all types of animals. And that is that they're undifferentiated. They don't look like they can do anything, and that's because they can't do anything. They're sat there waiting to go and divide and become other types of cells. So for that reason they have a large nucleus and very little cytoplasm, which is characteristic of all stem cells. And as you know, cells start to look different as they differentiate. So muscle cells and those have characteristic shapes and sizes associated with their function. Stem cells, when they're stem cells, don't have this. So these cells mean you can take an animal and cut its head off, as you probably did in class, and over the next 7 days you can watch it regenerate. So by 7 days it will have remade the brain, the eyes, and over the next 7 days, I'm not going to show that here, it will rescale to the correct size for the remaining tissue. Okay? Let's skip that. Right. So we use a particular species in the lab, called Schmidtea. It was found by someone called Schmidt, Mediterranean, because it was found somewhere in the Mediterranean. That's where the name comes from. It sounds more complicated than it is. And this species comes in two flavors. It has a flavor or a biotype that reproduces sexually here. And this is a cross-fertilizing hermaphrodite that lays cocoons, and these cocoons are polyembryonic, meaning they have many embryos in them. And when these embryos finish developing, the cocoon breaks open and you get hatchlings which grow up to be adult animals. The other type, on the outside, on the blue circle, is the asexual biotype. And this reproduces by fission, by sticking its tail down on the substrate that it's in, pulling, and leaving the tail behind. And this tail regenerates to make a new animal. And this head regenerates a new tail. And so this animal on the outside reproduces only by this method. Okay, so in fact these are now two distinct species. We think the one on the outside evolved from the one on the inside. Yeah, so the one on the outside lost the ability to make germinate cells and reproduce sexually. And so by definition, this animal on the outside, for it to survive over evolutionary time, must not age. Because there had been one animal originally that did this. And all the current animals out there in the wild are the offspring of that individual animal. So in a sense, they're all clones. Right? There's been no swapping of chromosomes going on here. They're just clones of each other. Okay? So if that's true, if you remember this picture of all the different causes of aging, there are many of them, not all of them. The ones to do with the immune system aren't, because these animals don't have complex immune systems. But many of them are relevant to this animal too. Except that this animal solved all these problems. So it solved the issue of stem cell exhaustion, it solved the issue of telomere attrition, it solved the issue of requiring mutations or epigenetic changes, all this other stuff that causes aging in us. And my argument, or our argument, with other people who work on this, not just me, is that by studying this really simple animal and seeing how it solved each of these problems, we might learn something really important. And that's what essentially we do. And that requires us to study regeneration, which most of you have been doing, but also the stem cells in detail, and also just watch them and see if they age, which is kind of an unrewarding task, because they don't. Yeah. A good question is, how can you prove they're immortal? I can't. Yeah.
How do you know if it's aged?
Well, you look for molecular signatures of aging, like telomere shortening or loss of stem cell proliferation, stem cells start to disappear. Okay, so I'm going to stop there with that part of the talk. Does anyone want to ask me anything about any of that? Yes?
What feature, so you know for the regeneration bit, like, you know the animals, what feature do they contain to allow it to regenerate?
What features?
Yeah, no, like, there must be something in order for it to be able to actually regenerate. So what's that feature?
Okay, so do you mean the information that tells it what's missing, or so the cellular basis is are the stem cells. So the animals have stem cells that can proliferate and make new tissue, and so that new white tissue. If I go back, so this up here is the regenerative tissue, And that comes from these stem cells that are dividing inside the body. Right? So progeny of these stem cells migrate, they make this new tissue, and that forms the missing structure. Is that what you asked? Okay. Yeah.
Do you think aging causes death, or something which happens in your daily life leads to death? Natural death, that is.
So you mean people who die of natural death in their sleep with nothing obvious wrong?
Yeah.
Okay. Well, probably when they do that, it's because there's nothing obvious wrong, meaning it's a general dysfunction. So normally when that happens to people, it means they go to sleep quite happy and they chat with their family and they don't wake up in the morning. And people who are old, that's probably because there's like a general dysfunction due to age. So their heart isn't great, their lungs aren't great, their liver isn't great, their kidneys aren't great, and then one day that all just comes together and they go over this kind of limit, and there's like this kind of a downward tumble of the systems in their body.